class now. We're going to learn about protein synthesis. So first I'm going to start with an explanation of DNA. So DNA is made up of a helix shape. Either side is a sugar phosphate backbone. This backbone acts as like rungs of a ladder, and then on each of these backbones it's got nitrogenous bases. So each of these bases will always partner up with another base. So adenine will always partner up with thymine, and that's what creates a code for genetic information. So when you want to replicate DNA, there's an enzyme called helicase that comes in, and it'll come in and it'll split apart this helix shape and unwind it so that you're able to access the information. So we're going to represent this with yellow as the backbone. So if this was your unzipped DNA, these are all the pairs of your nitrogenous bases. So this other backbone would come in once it's been unzipped, and this is your leading strand. So that's where all the information is going to come from, and it'll all be replicated. So these partners will all partner up, except the only difference is instead of thymine matching with adenine, we have uracil, a different chemical, and that's what creates the mirrored pairs. So from there we fill in all of these. And once they're all filled in, you'll have an entire strand of new information. This strand, the messenger RNA, will then leave the nucleus where it can be translated into amino acids. After the messenger RNA has copied all of these pairs, it'll go over to the ribosome. The ribosome is almost like a clamshell, and this string of information is fed through. As it's fed through, the ribosome transfer mRNA comes and reads the codons. So they're done in pairs of 3. So it recognizes first a start codon, in this case AUG, it's always AUG. And it knows to start copying this information. And while it's copying this information, the point is to match amino acids to these codons. Every protein in your body is made of amino acids. You produce, there's 20 altogether, 12 of them you produce, 8 you need to get through food. So once it reads this start, it knows to start forming a chain. So the transfer mRNA, it's like a clover shape, it'll come and read. So in this case, GAC, we'll say is this shape, and it'll come and drop off that shape or that amino acid, and then it goes away. So then as this section goes through the ribosome, another codon is read and another amino acid is dropped off. So as this happens, these chains are formed, these amino acid chains, until eventually it reaches a stop codon, and then that acid is complete. So in this case we've only used a few, but in reality these chains are like thousands and thousands of acids long. And then they'll leave the cell and create a shape, which is the protein that goes in your body and does whatever function it's supposed to perform. Sometimes this translation of information doesn't always go as planned. In some cases, the wrong information could be in the chain. So where there's a cytosine, it could actually be something else instead. And so that would alter the genetic information, but it doesn't necessarily affect the function of the chain or change it. It can sort of go unnoticed because it's a minor change. So a bigger mutation that can be caused by, say, carcinogens would be if something's completely added or deleted, because this would cause a frame shift change. So those sections of 3 that are read by the ribosome is now shifted over. So instead of reading GAC like it did before, it would now read GAG, and a different amino acid would come and be dropped off. And then from there, it affects every consecutive codon of 3, leading to larger mutations that can actually seriously damage or affect the cell.